Assalamu alaikum everyone. I am Dr. Sumreen Hayat from Department of Microbiology, Government College University, Faisalabad. I am also affiliated with Department of Biotechnology, University of Sargodha. First of all, I would like to thank to the organizers of this conference for providing me platform to present my work. And my research work is entitled as Aluminium Oxide Nanoparticles Inhibit EPS Production, Adhesion, and Biofilm Formation in Multidrug Resistant Acinetobacter baumannii. Due to emergence of resistance among numerous pathogens, it has threatened the scientific community, because clinical effectiveness of various drugs has been compromised due to emergence of resistance. When we talk about Acinetobacter baumannii, it is an opportunistic pathogen that is also a biofilm former. It is a superbug. It can resist multiple drugs, and a number of infections can be caused by this particular pathogen. For example, urinary tract, respiratory tract infection. When we talk about Acinetobacter baumannii, it can survive under in harsh environmental conditions in hospital settings because of production of exopolysaccharide and biofilm formation. So it means these are contributing towards the survival as well as towards the resistance against antibiotics. That's why in our study we mainly targeted exopolysaccharide production and biofilm formation in Acinetobacter baumannii by using aluminium oxide nanoparticles. And biofilms are microbial consortium adhering to living and non-living surfaces, and this community of microorganisms, it is enclosed by exopolysaccharide, and this EPS, as I have already told you people, that it acts as a barrier that will protect not only biofilm formers against antimicrobial agents, but it also EPS can also be used as a bridge between bacterial cells, our microorganisms, and surfaces to which they are attached. This is actually, this diagram is showing you various steps or stages in biofilm formation, and the process of biofilm formation can be divided into different phases like attachment, growth, maturation, and detachment. And when we talk about biofilm inhibitors, they can target any of these stages. And in our study, we mainly focus on process of inhibition of adhesion by aluminium oxide nanoparticles, as well as process of biofilm formation and pre-established biofilms. Either they can be targeted, they can be removed by using aluminium oxide nanoparticles or not. So this was basically the theme behind this study. When we talk about nanoparticles, these are actually nanomaterials, and nanomaterials are objects having at least one dimension in nanometer scale, that is around 1 to 100 nanometer. And the important thing about nanomaterials or nanoparticle is that they have high surface area to volume ratio, and due to this feature they have more they are biologically more active against as compared to their bulk counterpart. And another important feature about nanoparticle is that when we talk about when you want to compare it with antibiotics, that nanoparticles, they can target multiple sites in microorganisms when we talk about their antimicrobial activity, and as a result what will happen, microbes will face difficulty in exhibiting resistance against nanoparticles. So that's why today in current situation against multi-drug resistant pathogen, nanoparticles, they are being tested to find out either they are effective against them or not. In this study, we first of all, by using a wet chemical synthesis method, aluminium oxide nanoparticles were synthesized, and transmission electron microscopy and EDX was used to characterize synthesized nanoparticles, then antibacterial activity was determined by Broth microdilution method in order to check MIC and FBC. And then cell attachment assay was performed to find out the effect of aluminium oxide nanoparticles on adhesion to plastic surfaces that were provided in the form of microtiter plates. Later on, effect of nanoparticles on biofilm formation, as well as on inhibition of biofilm biomass of pre-established biofilms. It was also tested, and at the end we find out either aluminium oxide nanoparticles, they can affect EPS production or not by measuring the wet and dry weight of extracted EPS, as well as carbohydrate and protein content of EPS was also quantified. This is our result. Transmission electron microscopy indicated that nanoparticles, they were spherical or roughly spherical in shape, as well as diameter was around less than 10 nanometer. So in this diagram, you can see most of the nanoparticles, they are spherical. Similarly, EDS spectrum indicated the elemental composition of aluminium oxide nanoparticles, indicating that aluminium and oxide elements were present. Then the MIC for Acinetobacter baumannii, it was 125 microgram per ml, whereas MBC was minimum bactericidal concentration was found to be 1000 microgram per ml against all three tested strains of Acinetobacter baumannii. This figure shows you the effect of nanoparticles on adhesion of bacteria to plastic surfaces, and you people can see that when control cells were compared with treated cells, there was significant inhibition in percentage adherence to plastic surfaces. As you can see, inhibition in case of three strains was around 48 to 51%, and maximum inhibition was observed in case of Acinetobacter AB2 strain, where around 51% inhibition in attachment was observed. Similarly, biofilm, they were inhibited by nanoparticle. Biofilm formation was inhibited, like in the diagram, you can see the effect of nanoparticles on biofilm formation. Biofilms were grown for various time intervals, 24 to 120 hours, and in control cells, you can see that biofilm formation it was increased with the passage of time, and maximum was find out after 96 hours of incubation, and then the detachment process was started. Whereas in case when cells were treated with sub-inhibitory concentration of aluminium oxide nanoparticle, there was significant inhibition of biofilm formation when you compare it with control cells, and maximum inhibition was observed in case of strain AB3 at 48 hours. And then in the figure D, you can see the effect on preformed biofilms. Biofilms were initially grown for different time intervals, and then sub-inhibitory concentrations of aluminium oxide nanoparticles were added to find out either they are efficiently removing the biofilm biomass or not. And the results indicated that biofilm biomass was significantly reduced in case of treated cells when you are comparing it with control cells. Then at the end, we tested the effect of nanoparticles on EPS production, and the results indicated that in case of control cells, wet and dry weight was more as compared to that of treated cells. And it's the same thing was observed in all of these three tested strains. Whereas proteins and carbohydrates contents was also quantified, and the findings were that, like in case of treated cells, significant inhibition of amount of carbohydrates as well as proteins was observed. In most of the cases, there was significant inhibition or reduction in the amount of proteins and carbohydrate content was observed in Acinetobacter baumannii. So we can conclude that our study, the findings of our study was that aluminium oxide nanoparticles can be effectively used against Acinetobacter baumannii by targeting biofilm formation, adhesion, and EPS production. Thank you all for paying attention to my presentation.